Welcome, everybody, to the 6th episode of F.A.M.S, where I talk to friends, family, and complete strangers like Dr. Temple here about film, arts, music, science, and all the beautiful things that make the world go round. And so today, Dr. Temple is going to talk to us a bit about his journey from doing sound checks for Mitch Easter all the way to doing sonification of DNA of the coronavirus. So, Dr.
So check 1, 2. Check 1, 2 for science. Everybody there. Hey, so you brought up Mitch Easter's name. Yeah, I caught up with him about 10 years ago. He came to Australia for he was playing Big Star's Third. There was a touring band playing Big Star songs, and Mitch came out to Australia to play on that. And yeah, we met up for a few days and hung out just like the old days. It was nice.
Right. So for those of us listeners, or some of those listeners who are unaware or who've just never heard of a band called REM, or can you tell our listeners a little bit about your journey from your days in the Hummingbirds to where you are now?
Sure. So I think, like a lot of undergraduate, students, or just, young adults, I was always noodling in bands whilst studying. So I was a distracted student, to say the least, with my music activities. And at some point during the band versus science sort of experience of the undergraduate, the music won out. So I got to the point where I simply couldn't go and do my science work because I had gigs on and I had to go to Melbourne or Brisbane, other cities in Australia. And it just became too busy. So I gave up a Master's of Science, which was what I was studying at the time. I felt like a little kid joining the circus. I left my reliable science career to join a band, or the band that I'd formed, actually. So, and that was the Hummingbirds. So we put out a bunch of singles in Sydney, Australia, and this was back in the late 80s. So back then in Sydney, there was probably 8 or 10 venues within walking distance that had free music most nights of the week. We had Sydney radio stations playing Sydney bands. We had Sydney magazines writing about Sydney bands. And we had record shops in the city putting out singles by Sydney bands. So there was a whole scene happening, which made it very easy to begin a band and get a small audience and, and get things happening. So we sort of got on board with that and recorded some singles. And we managed to get them on high rotation on the radio before we'd even put them out on vinyl, just as cassette tapes. And yeah, so we got busy as a band. We recorded a whole bunch of independent singles, and then we sort of got coaxed by the major labels and had to make that decision, do we sort of shake hands with the music machine, or do we stay in indie band where we have complete control? And we decided to sort of go into the industry and try and make a commercial album with a commercial record label. And they were kind enough to ask us, who do we want to produce the record? And we had a few choices, and Mitch was one of those choices. And Mitch Easter, he's based in North Carolina, and we flew him out to Sydney, Australia to record our first album with us.
Wow.
And with our second album, we flew to Mitch. We went to North Carolina and record our second album with Mitch, where, at this really great recording studio. So, yeah, so I had a really good experience playing music and, like I said, joining the circus and leaving science for a few years.
It sounds like it's you can always leave science, but science never really leaves you, right?
Well, I think if you're in science, that there's a fairly clear career path which you can choose to take. And I think if you stick to the straight and narrow, that path is fairly well defined and there's lots of options. Well, I think in music, chances are rare. And when they come your way, it's pretty much a once in a lifetime opportunity. Yeah. So it made sense to give up what was a fairly, I knew science would be there when I finished my little excursion, but I couldn't choose when a record company was going to approach us and offer us, a deal, and we would just have an audience as we did at the time. It was a really great thing, really great experience to go through, being in a successful band. It was great.
And was this around the time you were working with the anti-cancer targets? And
yeah, no, I was doing a thesis on nuclear magnetic resonance structure of the Australian funnel web spider venom. So that was my day gig. And then I gave that up to join the band. And then the bands have a limited lifespan. I mean, not many bands go for more than 5 years, on average. So after our requisite 5 years, I left the band and went back to science. And then once I'd completed my master's, which I'd left, I then got a job in a lab. And I decided that the world of NMR was a very narrow, small field, and there weren't many people doing NMR. So I went to molecular biology, which was a much bigger area of research. And I started looking at, I joined a professor, and his projects was looking at anti-cancer drugs and their interactions with DNA. So I did my PhD on that. And then when I became an independent academic at Western Sydney University, where I am now, I set up my own lab, and that's when I started, carrying on that work with some chemists who were designing anti-cancer drugs. And then I was built a range of assays to see where those drugs bind to a DNA sequence, and then trying to work out if the potential new anti-cancer drugs differed significantly from the established cancer drugs in terms of their effectiveness.
I see. So life, unlike DNA, is definitely not linear.
Well, life is a circle. So it's like a plasmid. I always find I come back to the things that I thought I left behind, and I've done that quite a few times in life. I know some people talk about crossroads where you're going to make a decision. Where for me, the experience has been coming back to the things that I did before. So when I left science, I came back to science. And when I left music, I then came back to music. So I always seem to cycle through my interests and my opportunities.
I listened to the live performance that you performed with your mates, I think about 6 months ago.
This was the Exploding RNA Sequence Inevitable.
Yes, it was quite hypnotic.
I was being interviewed on the radio, and they said, "What's the name of your band?" And at the time, I was talking about Andy Warhol and his Exploding Plastic Inevitable, and I sort of reworked that title into an RNA experience, and then as a joke, we decided to use that band name at that little show we did, knowing full well that nobody would ever be able to remember the name of the band. It was a little bit, self-referential joke in that sense. But yeah, look, I'm joking about that performance. And for those who don't know, I got asked by by a government organisation called Inspiring Australia, and that they asked me to perform at this concert theatre in the city. So it's a full-blown concert theatre, and I was really excited to do that show. And I had about 3 or 4 months lead in to doing a show, and the idea of the show was I currently have a research focus on what's called sonification. So what I do, I take my interest in DNA sequences and the function of DNA sequences, and I use audio to read the DNA sequence. And then I try and use audio to help with the analysis of a DNA sequence. So I've sort of been using audio for DNA analysis. And obviously I'm a musician. So once I'd finished doing the audio analysis, I just had lots and lots of audio on my computer. And I'm scratching my head going, well, it's not music, but I think about it in some ways as music because it's got musical notes. And I designed all the mapping to say, these nucleotides go to these notes and these pairs of nucleotides go to these different notes and these codons go to another set of notes. So I had all this mapping of DNA data into ranges of notes and, you know, tried to make it sound, at least on my second attempt at sonification, I tried to make it sound more musical than the first attempt. For notes that I know are very frequent, I map them to like the roots and the fifths, that really tonal center. And then for the other things that are more diverse, I map them to other parts of the scale to just to build up the complexity over those basic root and fifth notes. So yeah, so I thought like a musician during my science experiment, which the first time I'd really allowed myself that luxury, if you like, because I was thinking, within my mind, I'm quite good at music and I'm quite good at science, but I never combined the two in a project. And I really wanted to do that. I really wanted to make, you know, good use of what I think I'm good at. And sonification seemed to be an excellent avenue for me to apply my skill sets. So to cut a long story short, I had all this science audio on my computer, and when I was doing outreach with this work as well, people were thinking about the audio as music. So even though it wasn't particularly musical, like, you know, science audio doesn't have an introduction, it doesn't have an outro, it doesn't have a crescendo, it doesn't have, you know, key changes, it doesn't have all of that stuff that we have in music. The science audio is this kind of repetitive beats, or, you know, these notes, these sort of percussive notes. And, you know, if you've got 6 billion bass pairs, you're going to have 6 billion notes. So, you know, your audio track's going to go for, I have never actually calculated it, but I'd assume it'd be for, you know, a year or two. You know, it'll just keep playing. So that there's none of that musical narrative exists in the science audio. And so what I did, I took the science audio and I took it into a recording studio. So because I used to be in a band, I've got a lot of music buddies. So I called on some old friends and said, look, this is what I'm doing. Do you want to come and have a, you know, have a play to this science audio? You know, so people were quite keen. So we got together and we had this do noise playing in the studio, which is the science audio. And I sort of started playing drums to that as if it's a kind of science click track, you know. So rather than having the my click track, I had this from the science audio. So I built up lots of sort of rhythmic sort of parts to the science audio. And then once I got those sorted, I invited the bass players and the guitarists to play to the percussive tracks that I'd built up to the science audio. And then we made this this range of actual musical tracks. And in the interim, I went downstairs, and this is what I dug out from a box. So this is a pressing of a record of the science audio with musicians, which hasn't been released yet, but it arrived in the mail all the way. You know, I only got 200 copies made, but yeah, so that's arrived. So at some point, I'm going to revisit this performance idea of the science audio with musicians and actually have a musical evening as a band would have, where we have a record launch. But it's a record that is funded by the School of Science, and it contains science data and musicians. So I really like the mix of notions of mixing art and science. And I was thinking where to launch the record, and I contacted in Sydney, we've got a national art school, and I thought it'd be great to have a science evening in an art school to really blur those boundaries between science and art. And the art school think it was a great idea. So I've I've got a space booked for August, and I'm going to try and work with some of the art school students to create some visuals, some digital art, whilst we play science music, and then try and have this sort of evening of science and art all mixed together into sort of a big mashup.
So it's possible there could be like a psychedelic ribosome, you know, just with the tRNA coming through and every code
on the walls. All of that being brought. So we're gonna try. I hope try and do broadcast on all the walls, like 360 sort of digital art, video sort of stuff while the band are performing. So again, it gets back to that, as you say, this sort of 60s psychedelia. Like I was never there; I was too young, but I heard Pink Floyd used to play at the UFO club in London, and it was these psychedelic nights with, we won't have the drugs, but we'll have the imagery, the art imagery and the crazy music. Okay, so I'm harping back to a tool I released in 2017. So this is my first attempt at sonification, and I'll play something that's quite repetitive. So I'm on my screen now. I'm just going to pick a telomere sequence, which is a telomere. Telomeres occur at the end of chromosomes, and there's 6 base pair repeats. So it's TTT AGG in some combination. So it's that sort of 6 base pair repeat. So if I'll sonify that now. Let me just see. I'm clicking a few buttons. All right. So what you can hear there is a da-dun, 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 which is the 6 base pair repeat. So it's very regular repeating patterns. But at some point, there's a small change in the DNA sequence. There's a 1 base pair mutation, and you'll hear that change because the da-da, 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 da-da pattern will change. So I'll keep playing it, and we'll see if we can hear that mutation. It's got a while to go yet. Oh, there it is.
Yeah. It's like a Morse code. It's like a Morse code of sorts for DNA.
It is. It is. I think I refer to it as a Morse code in one of my papers, thinking, you know, it's a way of, it's a different language, and it's not an easy language to learn. And I'm not suggesting anyone should be able to listen to a note and say, oh, that note is a methionine residue. I think that you could do that, but it would take a lot of learning to map in your mind the actual amino acid residues to notes. But what you can hear is changes in a pattern. And if you can hear those changes in patterns, then that tells you that in the sequence, there's a mutation there. So that was the idea behind that kind of mutation. When I first started looking at the things that other people had sonified, people had done the mapping of features in the DNA to codons and things, but no one had taken any thought process about, well, when you have a start codon, which is the start of a gene, why don't we have that as the start of the audio? So it's silent until that codon, and then that starts the audio. And the other thing that's really important for a biologist, but not important for a social scientist who does sonification, for scientists, we're acutely aware that DNA has 3 reading frames. So any sequence of DNA you care to look at can be interpreted in 3 different ways. And only one of those ways is the correct way to interpret it. So we have 3 reading frames, and the gene is an open reading frame. So I'm going to confuse everyone here and play a non-gene sequence. So the thing that you don't have a lot of in genes are stop codons. So in this bit of audio, a stop codon is a crash cymbal, and a start codon is a snare drum. And then if there's something to be sonified, you'll hear some notes. So you can hear there's areas of silence in there because there are stop codons in 3 different reading frames, and then there's nothing to sonify. So when there's nothing to sonify, you've just got to wait for that next start codon, which is that snare drum, or these other stop codons, which are those crashes. And this RNA sequence, which is a functional RNA, it's littered with stop codons. So it's untranslatable by the cell. So even just by sonifying these sequences, you can tell straight away what kind of DNA sequence you're looking at, which I think is really powerful, and I don't think, people who are very much visually centered on analysis have an appreciation of how you can generate audio to impart, information content as well.
This is certainly an additive tool to those that are doing data viz.
The very first time I went public with this sort of sonification was at an event called Making Science Beautiful. And it was all these people who had these beautiful art renditions of science data. And then I came up and gave my audio description of science data. And it was the sort of first time I sort of had the confidence to go out into the real world and talk about what I do. And just very quickly, I'll play you a gene sequence here. So this is the RAS gene, which is very important in cancer. And so what happens here is that you'll hear that there's one reading frame that is continuous. So on the other sequence, it's stopped all the time. In this one, because it's a gene, one of those 3 reading frames has to be on all the time. So you'll hear that when I play it. So you see here in the first reading frame, it never stops playing, but there have been stop codons to turn off the second and third reading frame. And at some point, you can see here, the second reading frame comes back on. So there's a start codon in the second reading frame, but it's the one in the first reading frame that is consistent throughout. So by way of continuing the conversation, so once I'd done the initial sonification, which is some of those examples I was playing earlier, it wasn't that musical. And I thought in the second time I did my sonification, I thought I'd bring in the musician part of my brain a bit more into the project. So I was about to start sonifying mitochondria genomes because it's just a small circular genome. And I thought that would be, you could listen to the entire sequence. But then I was in lockdown. So I changed from the mitochondria genome to the coronavirus genome because, it's 30,000 base pairs. It's nice and short. So effectively, you can listen to the entire sequence if you wanted to. It takes about 2 hours. But if you wanted to listen to a gene, it takes about, anywhere from 30 seconds to a bunch of minutes. In the previous work, you could only listen to one algorithm at a time. So it was very, it was very robotic. And music isn't robotic. Music it's more engaging than that. So I decided to do various algorithms, same algorithms as before, but I'd run them all simultaneously. So you'd be hearing, different algorithms processing the data differently, but they're all layering the audio into one audio stream. And here's an example, if you like, of the sonification of the coronavirus genome. I built a tool here, and I can explain the tool if you want me to. I've got a sort of an animated graphic at the top here, which tries to show these polymerase proteins running down the RNA as if the RNA is kind of like a railway track. And these proteins move along the railway tracks. And as they're moving along, they're reading the sequence and doing biological things with it. But what I'm doing, I'm reading the sequence and generating musical notes with the sequence. So here's an example of what it sounds like. So this is the protein here. If I click play. So, that just keeps playing this sort of quite melodic, because it sounds like a Game Boy. I got a lot of people saying it sounds like you're playing, Mario Brothers game.
Yes, it took me back to the Nintendo days. Yes, the early one.
Yeah, that's right. And I think that's a perfectly fine comment. if I reach out here, I can show you. Here's my Game Boy. I'm a big fan of the Game Boy. So I was really quite chuffed when people said it sounds like Game Boy music. I went, yes, that's really cool. But you know, but a bit more seriously, so in this one here, I'm trying to sonify the process of translation. So either of those three reading frames may or may not be a part of the audio. And I create a sort of a landscape audio just by looking at individual bases, which are always present, and pairs of bases that are always present, but codons that would be translated are not always present. So you get these layers and these textures building up in the audio. So if I go back to the beginning here, for argument's sake, and play this, you won't have any codons in this sequence. And the tempo is different because I change tempo depending on whether you're in an untranslated region or whether you're in a translated region, just to try and give it that sort of a more musical context. So this will play at a slower tempo than the previous example, and without those codons on top of the audio. And when it reaches the gene, it sounds like this. If I click here, when it reaches the gene, it sounds like this.
Wow.
So the tempo picks up, and then you've got that coding sequence, which is those letters on the screen. I don't know if you can see my screen, but those letters are added to the audio. So it's this idea of building up layers of audio, and each layer is a layer of science information, but it's also a layer of music that hopefully complements the previous layers of audio that were present.
It's like a song. You know, as producers know, you build layer.
It is, because when I sent the manuscript for the coronavirus work to the journal, the reviewers spent a lot of time and wrote some fairly lengthy, you know, discussions on it. And one of them was having quite a deep conversation about how it didn't have, or he was talking about it as if it was music, which I thought was interesting. I thought that was a victory. But he was also talking about the absence of musical themes. So one of the things I did as part of the review process was change the tempo of the introgenic regions compared to the codons. So you get these pickups, which is a bit more musical. It's this idea of changing tempo during a song, you know, which is quite common. Yeah. And then with layering these other things, I think that's quite musical, even though, you know, it serves a function in terms of relaying science information. So yeah, I thought it was really cool. So I have all these audio patterns. I've got a page here, which I'm sure you've looked at, which is just my sort of online bio, where I talk about the coronavirus music. And I'll give an example here of some of the audio that I was taking into the recording studio. So it's the same kind of thing I was playing from the tool. It's just, if you just listen to it here. So it's that same raw audio, but then when I take it into the recording studio, and this is the album I've been recording. If I play some tracks, a bit of this, you'll hear, you should now be able to pick up the sound of the DNA audio, because I've played it a few times, but then you'll hear the musicians start to play on top. And that's when I think it gets interesting. So here's one called Corona Code Music. So there's that DNA audio that we know, with a bit of hi-hat.
Nice.
So now we've got drums and keyboards and some guitar starting to build up.
And if I'm not mistaken, this is track 1, correct? On your album?
Yeah, that's track 1.
Yes.
Yeah. We actually came up with quite a lot of tracks. We came up with 12 tracks. So it was quite a productive period in that sense. And look, I worked with a bunch of musicians on this who I've listed at the bottom of the page here. So, you know, Matthew, Paul, Tim, Mike, and the Genome. And I was playing drums. And I've got to say that everyone got involved in the project for the love of the project, just wanting to be involved in the project. There was no, they got no money for getting involved. And they were so into what we were doing, and they came to every single rehearsal, and they all turned up at the live shows and gave these exemplary performances. It was really heartwarming from my point of view that everyone was so engaged and involved in the project. I thought it was a really great experience from a personal perspective.
Where can they find this or purchase a song?
So what I did, I put it on Bandcamp. If anyone wants to, particularly wants a copy, if they just write to me, I can send them, links to the music. They don't have to purchase it. It's not, I'm not trying to make money out of it. But I put a $7 price tag on there. I don't know. I feel a bit, don't know whether I should price it or not. It's a real question mark. But, it'd be nice to get some money to give back to the musicians. Because like I said, they came and they played out of the goodness of their heart. So I'm not trying to make money, but I would like to, give them a token,
Yes.
You go buy yourself a cup of coffee. And what's great for me is just listening to other people's contribution to the audio and the sort of rhythm tracks that I laid down. This one here, the science audio isn't as upfront, because sometimes I just think we wanted to drown out the coronavirus audio. We just wanted to smash it into the ground. We wanted to defeat the sequence, So this one starts off with a bit of Game Boy music, actually. There's that Polly A tail that you like so much. When I was doing the coronavirus sonification, I also sonified the process of copying the genome. So, and because the coronavirus genome is a messenger RNA, it's got a poly A tail. So the sequence ends with all these A's. So, and I kind of sonified those A's. I didn't really mean to, but when I listened back to the way I'd sonified it, it reminded me of the introductory theme to The Twilight Zone from the 1960s. Because it's got that weird dissonant kind of distance between the notes of the poly A tail. Anyway, so this is what it sounds like. So it goes from that repetitive sequence into a more diverse sequence. So when I took that science audio into the studio, we came up with this track here, which is, and I'll just play that. If you think back to that do-do-do-do-do-do-do-do that we heard earlier, you'll hear that when it comes into the track. You've got to wait a couple of seconds before it comes in. So you can hear how the science audio is embedded completely within the songs, and the science audio can be heard if you know what to listen to.
Right.
But I wanted it to be actual music that you can listen to without knowing anything about the concepts behind the music. Like if someone was to play this on an independent radio station, then I would want someone at home to hear this piece of music and say, oh, that sounds interesting. That sounds really a good cohesive piece of music. And then if you're a scientist or interested in science, I want those people to go, oh, I can hear the science data in that music. I can hear that stuff that's not music, but I can hear it in a musical context.
Yes.
So, I was quite cognizant, if you like, to try and achieve both those aims. I wanted it to sound good by itself without any background knowledge about what you're listening to, but I also wanted it to sound so you could hear the science audio in the music.
As a screenwriter, possibly we could see something in the future as we'll have a character maybe take their DNA and sonify their DNA, and that becomes their song. That people they identify with. Absolutely.
Like I was reading an article in there's there's a science blog site that I listen to, Codon Magazine. I was reading Codon Magazine, which is a new magazine started by a young journalist in New York. And this week in Codon Magazine, they talked about how it's almost possible now to get your genome sequence for $5. Okay, there's a new machine which companies can purchase, and then once they've got this machine, they can sequence a genome. I don't know how high quality the sequence is, but for $5, you can get a copy of your genome. Wow. So I really think in the future this is not something that I thought about when I started doing this work, but in the current context, it'd be nice to sort of have, what you can do is you can take a bit of your sequence, plonk it into the tool, and you can listen to your sequence. So I think if you've got something unique about your DNA sequence, you can listen to that uniqueness in the tool. And I think that would be really interesting. Have you seen that movie Gattaca?
I was just thinking about Gattaca just now.
Yeah, at some point he queues up and he goes to a little shopfront window and he gets his DNA sequence, and I was thinking that's actually becoming, even though it's 6 billion base pairs of DNA sequence, you get, 1,000 sheets of paper rather than just a little sheet of paper. But, this idea of listening to your own DNA sequence, I think is something that, might be a useful part of genetic counseling. If you've got a genetic disease and you're trying to understand your sequence and the implications of your sequence, then maybe part of the genetic counseling could be you can listen to the sequence, in a way that isn't scary science data. It's actually a more gentle introduction to what the sequence is and what the sequence does.
Right now I'm thinking about applications for sending messages through bacteria, right? So you can generate some sort of a sequence within a bacteria that would have certain codes for something, and that's how these spy agencies would communicate, for example. I'm in writing mode right now. I'm in sci-fi writing mode, so it's
it's getting quite common to embed DNA sequences within genomes. Like I think some of the more prominent scientists have put their little tags of DNA into organisms. I was thinking, a good T-shirt to have would be your name in DNA. Like if you think about the codon, like for me, I would need the codon. My first name is Mark, so I need an M-A-R and K. So I'd need methionine, alanine, arginine, lysine, I think would be the 4 amino acids, whose one letter code of those amino acids is M-A-R and K. So you could have, T-shirts with your DNA sequence on them that spells something, some secret code that only your friends would understand.
Well, there you go, Mark. You have some something to promote there in August at your upcoming gig.
I should do a DNA sequence that spells out Mark Temple's Corona Code music. You're just giving me a really good marketing idea.
It's fantastic. And so for those of us that would love to follow you on your journey and want to hear more about your work, where can they find you on the socials?
I have a Twitter account, which is @MarkTemp. I can send that to you if you want to include it, @MarkTemp. I do LinkedIn, but I can't for the life of me remember what my LinkedIn details are. I've got a WordPress site, which I just call, what's it called? It's just templemark.wordpress.com. So I've got a sort of an explanatory page there that talks about what I do. It's not updated regularly, but it talks about what I do. But yeah, so maybe that and Twitter, at mark.temp, I think it is.
Well, we've certainly come to the tryptophan, the ochre, the amber part of this podcast, and I have to say it's certainly been a pleasure talking to you and just learning more about this technology that exists and it is available for individuals to experiment with and test out their own DNA, and just it's a way of communicating to the public. in science.
Yeah, no, thank you for taking an interest in what I do. I know we initially said we'd talk for about 10 minutes and it's been probably an hour. So I think you've got quite a big editing job for yourself, but I've enjoyed talking, which is why I probably talked for too long.
I enjoyed it. I enjoyed this. All right. Well, thanks again, Dr. Mark Temple, for joining us on this episode of FAMS. And we certainly look forward to sounds of the future and updates on your album release and your performance. So,
thank you.
Thanks again.